Okay, we are in the last part of the basically the module of population genetics that I was going to teach. So right now we're going to go into the evolutionary forces that shape variation and sort of like violate the principles of Hardy-Weinberg equilibrium, and how this can be detected and how we can use this to then infer things that are going on at the population level. So I'm gonna start revisiting a bit of your homework. This is the question about this rare disease that was caused by a recessive allele. If 4% of the population develop this syndrome, what's the frequency of individuals in the population that carry exactly one copy of this allele? So here we are asking very specific things. So first of all, we know that then if it's recessive, in order to be expressed, it needs to be homozygote, right? So we have a recessive homozygote proportion of 4% of the population. So 0.04. That is our Q squared, which corresponds to the genotype frequency of the homozygote recessive. So if we like square root this, then we get that Q is actually 0.2, and then we know that P is going to be equal to 1 minus Q, because Q and P are supposed to add up to 1. So then we know that Q is 0.8. Then if we are asking for the individuals that carry exactly one copy of this allele, we are asking about the heterozygotes, right? So if we then know that heterozygote frequency in a population assuming Hardy-Weinberg is 2 times P times Q, then we know that the answer then is 0.32, or 32% of the population is heterozygote for this disease. So also for question number 2, then we have this scenario in which you genotype a population and you find the genotype frequencies below. Then we ask, is this population in Hardy-Weinberg equilibrium? How would you test this? Then we know that if we clear for P, then we know that allele frequencies here for P would be 0.38. Then we know that P plus Q equal 1. So we know that Q is then 0.62. So then this is sort of what we would get from Hardy-Weinberg. So then if we know now the allele frequencies in the population, we can also estimate how much, like what would be the expected genotype frequencies if we had Hardy-Weinberg equilibrium. And we can just basically plug them in and multiply them against the total amount of individuals in the population that we sampled. And then we know that the expected individuals with each genotype looks actually a bit different to what we observe, right? We see that then there is much less heterozygotes that we would expect. We have almost twice the amount of homozygotes for WW than we would think we would have actually in this population. So one way to see if this deviation from Hardy-Weinberg equilibrium is significant is to do a chi-square test. Then this requires you to go to basically the distribution of chi-square values and check if this is actually significantly different from the expectation. So you have observed minus expected divided expected, and you sum this up for the different categories that you have, and then you see which value of chi-square is basically the boundary or the limit where you would establish a significant difference. This is a statistical test. You don't need to solve it right now, but this is how you would look into this. So going back to what we are trying to discuss here are deviations to Hardy-Weinberg equilibrium's assumptions. So a bit of a recap, we are assuming that there is random mating, that we have large or infinite populations where there is no genetic drift. We also have no selection, we have no mutation, and we have absolutely no gene flow. So when we assume that we are going to break random mating, the opposite of random mating is non-random mating, then this happens through, for example, maybe processes like assortative mating. Um, Here, this type of assortative mating means that then you have a partner that is going to be probably similar to you in some way, for example. Here we have this beetle, sort of like yeah, this bugs that are reproducing in the study, and they found that when they were successful mating, they measured the female body, like the body length, and the male body length, and they established that, you know, when the matings were successful, it was when this proportion was closest to one. Um, and then in the unsuccessful matings, most of them were when they were actually deviated from this sort of ratio between sizes. This means that there is going to be a preference for this like insects to reproduce with other ones that are like more or less the same size as they are. So this is creating a pattern of non-random mating in this population, which then can lead to yeah, different like subgroups that are mating within a population, which have then different consequences. One of those consequences of assortative mating is speciation. Speciation by reproductive isolation can happen in two different ways. There can be allopatric speciation, which means that there is geographical isolation. Here, for example, you have two populations in different forests, but they are divided by this mountain range. Or there can also be sympatric speciation when you have reproductive isolation within a population, but this comes from other factors, like, for example, the example that we just saw about assortative mating. It could be that then there is this process in which individuals are going to choose a very specific phenotype that they're going to reproduce with, and they're not going to reproduce with any other phenotype that's present in the population, and this leads to this reproductive isolation. There is a really cool example here in the Heliconius butterflies. These butterflies are toxic, so they have developed this coloration to sort of like warn predators about their toxicity so that they are not eaten. However, it is easier for birds to remember, like, for example, just one pattern of butterflies that is actually toxic than many. So they have sort of like converged into this mimicry rings, which are groups of species that are going to have the same pattern to advertise that they are actually toxic and that they should not be eaten. So here we have, for example, like this sort of ecological function that is driving then this phenotype to converge. But then, for example, there are these magic traits that then take these ecological divergence and then also make it sort of a mating preference that is going to cause further divergence. And in this case, for example, we have two mimicry rings that are actually coexisting in the northern parts of South America and the lower parts of Central America. We see these two morphs of mimicry that are actually living in the same places. So we have Heliconius melpomene and Heliconius cydno. Then we see that then, you know, this coloration, like these differences in coloration have their own ecological functions. So then we have this coloration that is going to signal that this species is toxic. However, this other pattern has also evolved as a different mimicry ring, and it's still advertising that, you know, the species is toxic, but it has evolved completely different, and this is why. Um, when they saw that, you know, there's more were sort of coexisting, they wondered, okay, you know, if they are both advertising that they're toxic, then do you think that, you know, they might like interbreed? So for the experiment that they did was basically see the preference of the butterflies to breed with another male, well, with a female that either belonged to their same color morph or to the different color morph. And what they saw was a clear difference that, that this, for example, Melpomene would try to breed with the orange morphs, while Sidno would also try to breed only with the white morphs. So this means that this sort of convergence of phenotypes that are going to advertise that they're toxic is also a trait that then is going to create non-random mating. Then the consequences of like breeding out of this like color morphs is that you're going to lose this coloration, like get intermediate values or some other consequences that that could sort of throw off the phenotype that is actually protecting the butterflies into something else, and that's why then this preference has a mating importance but also an ecological importance. And this is when these two things align in some traits, then this is called a magic trait. Then there is this paper that is really interesting that actually makes the argument that magic traits that lead to speciation are not really that magic, but are also not rare. And this, well, I forgot to tell you, but this example of the different color morphs that are coexisting in the same populations, in the same places, are an example of, for example, sympatric speciation. So we see how this can lead to two different processes, right? Yeah, and this leads to speciation without the need of actually having geographical barriers in there. Then another case of non-random mating is the opposite pattern of assortative mating, so non-assortative mating, which is the preference for partners with different phenotypes than yours, and this could also lead to different evolutionary consequences. So this is actually a really cool study that they did in humans, where they tried to genotype the genes of the MHC complex, of the major histocompatibility complex, which is a complex of proteins that basically has immune functions. And the more diverse these genes are, the more covered the person is against different disease or different pathogens. So when they did the study, they basically got like the odor of sweat of different men, and they gave it to different women to smell and to then rate like their preference to different smells. So what they found is that the women were choosing the more pleasant smell to come from men that had similar MHC type genes rather than having similar to them. This means that they were actually selecting for men that had genes that were different to hers. Then in the second sort of trial, they asked them, does this remind you of relatives? The smell, and then they couldn't really tell. But then when they actually asked them if it reminded them for a partner or an ex-partner, they could actually then say, yes, this smell that I prefer to reminds me of my ex. And then they could reinforce that this was actually a pattern that was driving basically mating preferences in humans. So this is one really cool example. It has been greatly debated, but then the evidence for it is in there, and we have then observed it in many other species that also drive their mating preferences to maximize the diversity of this kind of genes. A bit of a question here. Which type of non-random mating would you expect to increase homozygosity? Either assortative mating or non-assortative mating. So think about it for a little bit. Pause your video if you need to. But basically, if you are expecting homozygosity to increase, then you're probably going to find that when there is assortative mating. This means that you are selecting for individuals that have your same phenotype, and probably those phenotypes are going to be produced by the same genes that you have, and then the chances of your offspring having those same genes coming from either parent is going to be higher. So when you have assortative mating, you are going to have a higher homozygosity. So if you go back to this example, like the homework exercise that we left you, um, you can see that there is a higher amount of homozygosity than what you would expect on both dominant and recessive alleles, and then there is a lot less heterozygotes than you would expect in this population. So it might be the case that if you see this deviation from Hardy-Weimar equilibrium, it is actually due to assortative mating. So these are some of the things that you can start thinking about when you see how populations start to deviate from this idealized model. And basically, we have another force, which is, it starts to get a little bit more wild, but it's sexual selection. And here, for example, we see a very extreme example of how males um, start developing traits are going to be specifically destined for sexual selection. For example, the tail of the peacock is the prime example of sexual selection, and it also opens this paradox. Then, is sexual selection, increasing the viability of the species, or is it just going to sort of be like a test of how selfish I should be with how I choose my partner? Because, for example, if you see this huge tail, you can probably think that it's going to impair the peacock of flying a lot, or it's going to be harder to run away from predators. So then this is a paradox that is still really debated. Is sexual selection really aligning with natural selection, or is sexual selection doing something completely different? But that is also a force that is going to significantly affect how populations evolve. And these patterns of non-random mating then actually create reproductive skews in animal populations. So this is a study in crystal frogs. I think the genus is Espadarrana. And you can see here there is a like the population, the frequency of individuals that had different number of mates. So, for example, there were really very few males that had a lot of mates, but then I would say that is almost 50% of the population that actually didn't mate at all. So when you have this, for example, forces of sexual selection, or you have this like assortative mating or non-assortative mating, actually the number of individuals in the population that can reproduce is going to be decreased. And this takes us to then trying to understand actually what effective population size is. So effective population size is going to be very different to census population size. So for example, here, take the same frog population. We have a lot of individuals, but actually the ones that are contributing to the gene pool are only very few. So imagine that it's these 4 individuals that are contributing mostly to the gene pool in this population. The actual effective population size, assuming that you have an idealized population where everyone has the same chance to reproduce, is actually very small in comparison to the census population size, which is much bigger. It can be that there are several factors influencing this, but one of them is, of course, sexual selection. So effective population size then is the size of the idealized population that you would expect to have the same effect of random sampling and gene frequency as in the actual population, or how you would expect a population of different sizes to behave if all of the assumptions of equilibrium would be met. However, here we are violating one of the main ones, which is infinite population size. So if you start having a lower and lower and lower population size, then strange things can happen. So again, we are now moving to the assumption of large populations, and here we are going to introduce what genetic drift actually is. So genetic drift is a change in allele frequencies due to random chance. Here, it can be defined as an artifact of sampling in a finite population. But it's easy to imagine, for example, that if you have a population of 10 million individuals and one dies, the allele frequencies are not going to change that much as if you, for example, have a population of 10 people and then one of them dies, and then basically 10% of the population disappears, and with them their alleles, and this is going to skew a lot the genome, like the allele frequencies that you're going to have in this population after this event. So for example, take here a population of balls. Half is red and half is blue. Just by chance, you grab a given number of balls from this jar, and you end up with actually having a lot less red balls than you had, because you were just sampling randomly. Then it could be that just by chance you end up, after the several sort of sampling genotypes in the population, to end up with a population that has only blue balls. So this establishing that a population size in the Hardy-Weinberg equilibrium gives rise to the Wright-Fisher model. And then the Wright-Fisher model is basically having all the same properties as a Hardy-Weinberg equilibrium, but we are establishing a limit for population size. So for example, here we have a population of these different colored balls, and we are going to randomly sample, assuming that there is random reproduction to the next generation. There is no mutation, there is no gene flow, there is nothing else than just a finite population. Then we select the blue, we select another blue, then we now select the red one, a blue one, another blue one, and so forth. And here, as you can see, there is already a change in the population composition, despite there being random reproduction, despite there not being any other evolutionary forces in play. So this can happen again and again until you end up with a population that is completely blue. And this is basically the process that then we can use to sort of calculate the probabilities of a population then coming from a single individual, or having a trajectory of the population that is going to sort of go back in time. So what is the chance that we end up with a population that looks like this if we go all the way back to a single individual? Basically, this is a very interesting way of imagining how population processes actually happen, because then it is basically all based on probabilities of one generation passing their genes to the next, right? And this is going to be highly dependent on population size, and basically, if you derive mathematically this model of coalescence, then you see that the time it takes from the past to the present, well, basically, if you are simulating to the past, the time it takes to coalesce to one single individual, it's usually the effective population size. In the number of generations that it's going to take is actually equivalent to the effective population size at the present time. So this is a really interesting way to model how populations behaved in the past, and basically what is the probability of a population to look like that in each generation, and then finally finding this last common ancestor of the entire population. So as I was telling you, here we have a representation of the effective population size on allele frequencies, for example, here in P, over time. And we have, for example, different initial gene frequency, allele frequencies here. And we see that these are going to behave pretty erratically. There is no selection, there is no mutation going on around here, but we just have different initial allele frequencies, and this is how they are going to behave over time in a population, for example, of 50 individuals. And all of these fluctuations come from this sort of like artifact of genetic drift, like each time that they are, selecting for a new generation or like passing genes randomly to a next generation, you see these fluctuations in allele frequencies. That means, for example, that here if the allele is really rare, most of the times it's going to go extinct, but sometimes it might just drift to be really common in the population. And here, for example, in 0.5, you have almost basically a random behavior where the allele might go to fixation or it might be extinct in the population without any forces other than population size. So when you increase population size, what you see is that this behavior is not that erratic. You start seeing that genetic drift is going to be a much more influential force in small populations than in larger populations. And this is how then we can sort of imagine like an idealized, really large population where this is not really happening, where this really small random events are not going to have a bigger effect in the population. Then, for example, for me, this is really an interesting topic. Because I work with conservation genetics, and I've always tried to figure out what is really happening in population bottlenecks. So basically, what a population bottleneck means is that we have this really large population of individuals, and their genotypes are represented as the little balls here, and they are different colors. Then we have a population bottleneck where only a few surviving individuals remain from a drastic reduction in the population. It could be a catastrophic event. It could be that their habitat was destroyed, or their habitat was like completely isolated from the large population where there was a lot of gene flow coming from. And then then they start reproducing, and then the population starts recovering in numbers, but not really in genetic diversity. So this leads to a low effective population size, and then because you have lower genetic diversity, you also have a reduced adaptive potential in these populations, because then we know that evolution is acting on variation, and if there is really low genetic diversity, and there is not really a lot of variation that is going to help you adapt. For example, if the environment is going to change drastically. This effect of reduced effective population size can also happen, for example, if there is colonization of an island or a new environment. So basically, you have only a few individuals that then go to this new area and they start to populate it, but then their genetic diversity is also quite small, and not all of the original genetic diversity from this ancestral population is going to be present in the new population. So then again, you have a reduced effective population size here. This is a sort of like simulator in which you can sort of simulate the different effects of genetic drift, and you can play a bit with population size, number of generations, and original gene frequencies, and you can see how they actually behave. You can play with different parameters, and we can include this simulations in the link in Slack. And also, I think I will share with you some resources that I found really cool, or you can also exercise a bit of your R skills while you go through like some practical examples of this kinds of simulations. Then we move on to the next assumption, which is no selection. And here we introduce the concept of selection in populations. So, for example, here we see that there is a distribution of the phenotype in a population. So for example, in this case, fur color, and you're going to have a normal distribution to start with, and you're going then to see how this is like how the frequency of these phenotypes is going to vary in the population, right? So if you see, for example, that then this brown, sort of like the middle color morph, is starting to become more and more frequent in the population, then you can see stabilizing selection. So it is going to eliminate extreme values of phenotypes, and it's going to narrow the width of the distribution, because then this is the phenotype that is going to better match the environment, and it's the one that is going to be selected for in this population. Then, for example, we can have directional selection in which a tail of the distribution is actually selected for, while the extreme values at the other opposite end of the distribution are going to be disfavored. So this is going to actually shift the distribution of the phenotypes to the darker phenotypes, because then, for example, again, you could have like the classic example of the pepper moth, that the darker phenotypes are going to actually blend better with the environment, and they're going to be selected for. So that is an example where you have directional selection. Then, for example, you can have the distribution of phenotypes to actually diverge into the extremes. So, for example, you have a very light fur color and very dark fur color coexisting in the same population. And this could be, for example, because there are parts of this population where there is I don't know, the soil is actually quite light, and then you have other patches where it's quite dark, and then this both phenotypes can actually escape predators better, for example, than if there is a brown fur individual that is going to be visible in most of the territory, right? So then you're going to have this disruptive selective force that is going to correspond to the pressures that you find in this population. Then when we are trying to quantify selection, actually, then we are going to go with this notation for the different genotypes, and this is going to be actually a notation of fitness. So what is fitness is really big kind of words, but we're going to assume right now that it is a number. And this number means that you are quantifying basically how successful your genotype is going to be in a given environment. And this can be, for example, quantified in number of offspring that are successful, in number of seeds produced, any different ways, but right now we're going to just assign numbers. So for example, here we see that then the homozygote dominant genotype is going to have a fitness of 1, the heterozygote is going to have a fitness of 0.98, and the homozygote recessive genotype is going to have a 0.92 fitness. So then how does this generation is going to look like? We are going to adjust then the values here to the frequency of each genotype. So here we're going to be able to quantify basically the relative fitness of each genotype by dividing its fitness times the frequency of the genotype in the population divided all of the well, basically the average fitness of the population. So then this mean fitness is going to be the way that we scale how all the genotypes are going to be selected for in the population, and basically this comes from multiplying their fitness to their frequencies. So basically what we can see with stabilizing selection, for example, imagine we have a fitness where the heterozygote is actually doing better than either homozygote, then regardless of where the frequency of big A starts, for example, we can see that it is going to converge into an equilibrium frequency of 0.518, because then we have this selective forces pushing to have both alleles in the population, and this is going to be a stabilizing selection. So here we have a different example. Here we have disruptive selection, and we see that then the fitness of the heterozygote is actually lower than both the homozygote genotypes. So these homozygote genotypes can then, for example, correspond to either phenotype in the population that is in the extremes of the distribution, and then we're going to see that, for example, if the frequency of big A is over 0.5, this allele is going to actually go to fixation, whereas if it is lower than 0.5, this allele might actually go extinct, because then we're going to have little a being the fittest allele in this type of population. Despite having the same fitness in the genotype, the initial allele frequencies are going to actually then decide the fate of either of these alleles for this gene. So then if we have, for example, the recessive, like the dominant homozygote, with a fitness that is higher than the heterozygote and the recessive homozygote, we're going to find that then this big A is going to basically go to fixation. But when we observe that then the recessive homozygote has a higher fitness than either the heterozygote or the dominant homozygote, we are going to see that even if the big A allele is really sort of like common in the population, it will slowly decrease until it goes extinct. This is because of how we've coded here the fitness of each genotype. And this will correspond to directional selection. So we are selecting for one of the extreme values, and we know that then this genotype is doing actually better than any other genotype in the population. Now we go to the next assumption, which is no mutation. And this is also an interesting avenue that we then go into. Then explaining evolution is actually a key part of how evolution actually happens. So mutation is going to be the process in which there is a generation of genetic variation over which natural selection is going to act. So this is really the key part as to why microevolution keeps on going. But then not all mutations are the same. So then mutational effects can be either neutral, deleterious, or advantageous, right? When you plot the distribution of how, what are the effects of these mutations, for example, here we assume that fitness effects that are neutral are actually going to stay in values around 1. If the mutation is deleterious, then they're going to have values below 1. And you can see that there are a lot of mutations that actually go all the way to 0. This means that these mutations are actually lethal. So we have a lot of mutations that are very bad for you. We have certain mutations that are going to decrease your fitness a little bit. Some mutations, usually like synonymous mutations, that will not have any effect, neither deleterious or advantageous. They're just going to be neutral. But then there is a small proportion of mutations that will be advantageous to the organism, and this is going to be actually selected for because it's going to give the individuals a higher fitness than the ones that don't have this mutation. And one of the tools that people use to sort of like then assess what's going on in populations and how these mutations sort of arise and how they should behave in certain populations is the site frequency spectrum. So for example, we have here the number of sites that are going to have different alleles. However, we see that for most sites in the genome. So basically, all the base pairs in the genome have a site in which we can compare if there are different base pairs or not. We can see that most of them in populations have the same base pair there, and this is actually fixed in the population. So there are no derived alleles, and there is there is basically no other sites, or there's no sites that have this other allele. But when we find sites that are actually polymorphic, meaning that they have different bases or different alleles in these sites, we can then have sort of an expectation of how this will look like. So when mutation arises, they are going to be around in the population in very low frequencies. So if we imagine frequencies here in the x-axis, lower frequencies in the population, are going to probably be very, very new mutations. And there's going to be a lot of sites that are going to have this very, very rare allele. But then there's going to be sites where these rare alleles are much more common in the population, but there are going to be much less sites. We are expecting to see this sort of curve here, this pattern of going with very new mutations that are probably happening a lot, are very recent, but not a lot of the individuals in the population have them. And then if mutations manage to persist in the population for some time, they are going to hang around in a lot of individuals in the population, but there are going to be much less sites than what we see for really new mutations. This is basically due to drift or to other processes in the population, and these new mutations don't really catch on in the population and they might disappear. But then it's only a few that actually make it to increase their frequency in the population. So we can see then what happens when this sort of pattern is broken. For example, here we see that then there is a shortage of rare alleles. Then these rare alleles that you would expect to have are not really there anymore. And the reason why is that there could be a bottleneck event. So then, for example, if you have this random disappearing of individuals or this catastrophic event, you can imagine that the alleles that are going to actually survive are the ones that are going to be more common in the population. So then you have sort of a shortage of rare alleles, and then the different polymorphic sites that you're going to have are more common in the population, are probably going to be there once the bottleneck is gone. So yeah, this is a way that people have even used to detect bottleneck events. They expect this distribution of rare alleles in the population, and if it's not fulfilled, then you can say that the population is going through a population bottleneck process. Another way where this can actually happen is when you have balancing selection for some sites, for example. So when you have these new mutations, they're not going to behave like neutral mutations, so they are going to actually increase their frequency quite quickly, because then these new mutations are actually going to be favored, and when there is balancing selection, because then you have more diversity, like for example in the MHC genes, that are going to be favored, whereas for example other kinds of selection might favor one specific allele over another one. So here you are going to have for example every new mutation that comes that might be beneficial is going to be quickly increasing the frequency in the population and basically being more present in more individuals. Then, when you have an excess of rare alleles, which is basically the other side of this pattern, you are going to probably be experiencing a population expansion or selective sweep. And what does this mean? Then what you can expect then is that if the population is expanding, there is a lot of recent reproductive events. There is a lot of individuals that are really recent to the population, and you expect this new mutations to still be around and not really have that be drawn out by drift, or having been pulled by selection to be actually very common in the population. So then you have a lot of these really recent mutations, these very high richness of derived alleles, of rare alleles are going to be in the population, and this is sort of a hint that the population is expanding. But it could also be that there are selective sweeps where you basically have this new mutation that is sort of bringing all the other mutations around it to a higher proportion in the population. Then you are going to find an excess of these rare alleles that are just sort of like hiking into this other individual, and they're persisting in the population, they're increasing in the population because of the selective sweeps. Then the last one, the last assumption is that there is no gene flow in these populations. And then this is a very, very basic assumption in which there's basically no interchange between individuals of other populations that bring other genetic material and reproduce with the individuals of the population that you are observing. So for example, here, you have two populations that have been evolving separately, and then this geographic barrier, for example, is eroded, and then you have a path that allows then the influx of individuals from one population to the other. You can see, for example, that these two populations have been evolving in isolation for a while, and then there are probably alleles that got fixed in the different populations, and they are likely to be very different alleles. So when this happens, you have an admixture event between both populations, which means that then you are going to have this new allele coming into this population, which is in a way sort of a mutation event if you see it in terms of allele frequencies in the population. But then this is again going to change the allele frequencies in this population and is going to cause evolution, right? So there are different scenarios in which you can represent gene flow between populations. For example, this is a meta-population level where migration is noted as M, and you can see here that there is gene flow among all of these different populations, and then you can probably vary the rate of migration M. So when there is less migration, these populations are going to be more different because they are evolving separately. But if this M is really high across all populations, then you can expect this sort of system of populations to behave as a single population and have a lot of mating and gene flow going around. So a measurement that is used to describe the gene flow or the structuring of populations is actually FST. And FST basically calculates what is the divergence between these populations in terms of their allele frequencies. So there are very different models in which you can sort of simulate migration. You have a sort of like another meta-population system where there is one middle population that then connects to other ones. You have like a main population that has this smaller like satellite population, where there you can have this sort of like ring of populations that are allow only sort of like gene flow between them, but not really across all of them. And this is going to give you different patterns of gene flow. Different, for example, these two populations are going to be much more related than this to this one. And then this is how FST becomes useful. So for example, here you can expect FST to be very low because there is very low divergence. There's a lot of gene flow. This population is basically behaving as one population. So FST is expected to be low. However, in this case, in this last case, for example, the two populations that are adjacent are going to have a much lower FST than, for example, ones that are like completely opposite to them. Imagine like this is a mountain range that is round, and then you have these populations that are restrained by height or like altitude, and then there is only like horizontal gene flow going around, and you cannot really cross over the mountain. So then these populations are going to have certain level of relatedness, but then these ones, like the ones in the opposite ends, are going to have a lot of divergence between them. So this is basically the end of the population genetics module. I hope you liked it. Um, we went over a bit of how populations evolve in the macroevolutionary scale. Thanks to the ideas of Darwin and Gregor Mendel, we had the chance to create this modern synthesis of how population genetics occur and how this also leads to evolution and how evolutionary forces are going to sort of deviate from the Hardy-Weinberg equilibrium that we have observed and actually drive evolution not only at the population level but also at the macroevolutionary level with speciation and biogeographical patterns of how we then perceive animal communities, plant communities, and entire ecosystems. Then we know which are the specific forces that are going to act over these populations over time and are going to lead to change, are going to lead to evolution, and then we can actually quantify them as deviations from Hardy-Weinberg equilibrium. So this is, again, the importance of how we can develop a whole field out of a known model, and how we can actually quantify what is going on in population genetics. And again, like give you all of this with a grain of salt, because then population genetics is actually a very, very theoretical field in which there are a lot of assumptions. Probably the biggest one is how simple the genotype to phenotype map is. But then this is a really, really important tool and really a powerful tool that helps us understand how populations and how organisms evolve. So thank you for making it all the way here, and I'll see you in the Q&A sessions.